Hello, Bio 4. Yay! This is our absolute last section. This is your last set of lecture slides that you need to go through, that you need to make sure you understand and know for the rest of your assignments that are coming up. And this section is called the control of plant growth and tropism. We have to understand how a plant will grow and how tropism works and how the plant will respond to external stimuli. So that's what this section is about. And quite frankly, I want to just jump right into it. It's not a very difficult section. We've had worse. So I think that this is going to be a nice section for you to just round off this module. So the first thing that obviously I want to start off with is we've got to understand some general points about plant growth. We've got to just understand how it starts, where it starts. You're currently looking at a paper about germination and you're understanding when a plant is considered germinated. So we've got to understand plant development, and plant development consists of three parts or three phases, and that would be embryogenesis, so the actual formation from the seed out into a seedling, the vegetative process, so that growing phase, that teenager phase almost, and then the reproductive phase, whereas it's going to reproduce its own offspring. So we have to understand embryogenesis, vegetative growth stage, and reproduction. We are going to touch on embryogenesis just very briefly. You just need to know what I present to you guys. The vegetative stage is where we're going to be focusing most of our time, and that's going to be focused in on those meristematic regions that we love so much, that we were introduced to in our first set of lectures, and then we are not touching at all on reproductive stage. There's just too much of those three stages to actually cover that is not within the scope of this module. So just bear that in mind. We're going to briefly discuss embryogenesis. Just, this is what it is. This is how it works. This is the different stages. And we're going to focus in on vegetative growth with our meristematic regions. We're not even going to go into hectic detail about that. Just to enough for us to understand the meristematic regions and to understand how plant hormones work. And we're not going to touch at all on the reproductive stage. But you need to understand that plant development has three stages: embryogenesis, vegetative, and the reproductive stages. When we are discussing the vegetative growth stage, which is essentially the growing stage, we have to understand tropism, because remember that the tropism will determine in which direction the plants will grow. So it's a very important phase of the reproductive growth system, and we have to know it. So first thing, I would like to just get through some basic concepts. We need to just we need to understand basic principles of plant growth, which we have already. I want to just quickly compare the development in animals and plants to specifically that vegetative growth stage, the importance of hormones. Now remember, when we look at tropism, when we look at the growth of the plants, a lot of this is going to be dictated to by hormones and genetics. Hormones and genetics. Remember that. It is absolutely important. And then plant tissue culture. Plant tissue culture is where my main focus for my PhD is in, which is growing a plant in aseptic, so completely sterile environments, no fungi, no bacteria, no germs, and seeing how plants grow on agar, which is a alternative to soil. So you might want to consider reading the chapter from Hopkins. It's on Moodle, so it's already there for you guys. But ultimately, plant tissue culture is using small pieces of plant tissue on a nutrient medium under sterile conditions. So growing a plant under sterile conditions on a nutrient medium. Okay. So here we go. Basic principles. Remember, I've mentioned we've got three phases: embryogenesis, vegetative, and reproductive. So we need to understand this idea of a zygote to embryo to adult. And if you remember, you will you know that we start off with a zygote and then an embryo and then an adult. We need to understand that embryogenesis. Remember, embryo, all sides divide, and then we're later going to get our meristematics. Please remember that the apical meristems are determined during embryogenesis. So when that seed starts growing and it starts getting into its different stages, which we'll see later on, you have to understand and be aware that those apical meristems, your main meristems, your ram and your sam, are formed during embryogenesis. The other ones, the secondary meristematic regions, your lateral and your intercalary meristems, are formed post embryogenesis. So when you already have a seedling, that is when they will form. So in embryogenesis, we have different stages. There are four: the globular stage, the heart stage, torpedo, and maturation stage. What you need to understand is that the meristematic regions get created during different stages of the embryogenesis, but only your ram and sam, only the apical meristematic regions, the ram and the sam, are formed during embryogenesis. So these interdeterminants, which are permanently dividing, they are permanently apical meristems at the bottom and at the top. They are formed during embryogenesis. The determinant, the lateral and the intercalary meristematic regions, which are determinate, which they produce a specific organ, a leaf, a flower, a node, a fruit, etc. They are formed post embryogenesis, after embryogenesis, when we've already got an established seedling. So embryogenesis consists of four stages: the globular, heart, torpedo, and maturation. It is somewhere in between embryogenesis that we get our apical meristemats forming. When we have a seedling already, so I'm going to hop to the next stage. When we have a seedling, this is already a mature plant. When you have a young seedling where the two primordials are still forming, it is then that we're going to get our secondary meristems, lateral and intercalary, which will start with secondary growth. Remember, primary growth, secondary growth, we've covered this. You are aware of all of those terminologies. So your apical meristematic regions are in charge of vertical growth. Your lateral meristematic regions are responsible for secondary growth. Remember now, interdeterminate, that RAM is permanently dividing. It's indeterminate. It's going to tell what the cells need to be at later stages. Those determinate meristematic regions, they know what they're going to be. They know they're going to be a leaf. They know they're going to be a secondary stem, etc. So they are post-embryogenesis. Okay. So you need to be aware. You have to take away what was learned in the first set of slides about RAM, SAM, and the secondary meristematic regions, and now you need to add on the fact that the apical meristematics form during embryogenesis, and the secondary meristematic regions form post-embryogenesis. You have to be able to make that connection between the first set of slides and this last set of slides. So if we want to just quickly compare comparison of animals and plants, remember that animals have a fixed amount of limbs, fixed amounts of body parts, fixed amounts of things. Of course, you're going to get damage, or you're going to get mutations within the genetic code sometimes to get extra limbs, etc., or no limbs, or whatever the case may be. But generally, animals are fixed. They have their genetics have predisposed them to having a certain amount of legs. So the genes, remember, the plants as well are governed by their genes. So for animals, we have a predisposed genetic makeup telling us how many legs we're going to have, whereas a plant, it doesn't matter. You know, it's plastic. It could be 2, it could be 3, it could be 4. However, our genetics also, the genetics still governs the plants. So a specific species might have between X and X amount, whereas another species might have a little bit more on average or less on average. But just because you're this species doesn't mean you're going to have X amount of things. It's very plastic. It can change. Remember, plastic phenotypic plasticity, it can change. The morphology can alter. So it's not set in stone. Just because I'm part of this species means I can have 3 branches or 4 branches or 5 branches, but my genetics doesn't allow me to pass a threshold. Remember, the genetics set the threshold. Remember, animals, we have a cardiovascular system. We also have a nervous system. Plants, no circular system and no central nervous system. That in no means the plants don't feel. Hello, vegetarians. What? Yes, plants can feel. Just because they don't have a central nervous system or a brain to interpret the pain, they do have hormones. And that's what we're going to look at. We're actually going to look at how a stimulus is going to affect the plant. The plant is going to produce messengers or signaling, and ultimately the plant is going to interpret it. Okay, for example, I'll give you an example. Everybody knows acacias. Those are those flat trees. They grow everywhere in South Africa. They've got those thorns. Very much the savanna kind of a vibe thing. But what these do is they actually communicate with each other. So a giraffe will come along, or a herbivore who can reach their leaves, and they'll start munching on the acacia leaves, nom, nom, nom. And the acacia, because it's being ripped apart by a herbivore, it reads that stimulus. Receptive cells receive that stimulus. They transduce it, or they send it to the signaling messages, and a hormone is released. And that hormone that is released tells it to release a pheromone, or release it's a pheromone, and the pheromone goes into the atmosphere through the stomata, and the wind blows at all the acacias that are downwind, when their receptor cells pick up this pheromone, they send it through the signaling transductase phase or process, the transduction phase, and then they respond by releasing a chemical into their leaves. So when the giraffe is like, oh, I've had too much of that, and it goes to the next one and it tries to eat it, it's actually yucky, it's chewy because it's released a hormone, or it's released chemicals that have made the leaf not palatable to the giraffe. So, just because a plant doesn't have a central nervous system does not mean it does not feel. It still produces ROS, it still undergoes stress levels, oxidative stress, and it still responds to that. It has an antioxidant system. It has an antioxidant process where it's supposed to balance the excessive ROS production. So plants do feel. They don't have a brain that tells them this is pain and we need to cry because we've caused ourselves a paper cut. But they do have, hey, we've been damaged, we need to reroute the water because some idiot took an axe to us, and now we've got conduits that are broken on that side, and he was just having fun, and now he's left, and we've got half of our conduits on one side is not able to be used, so we need to redirect. So they have an ability to respond. They have this ability to receive the information, to transform the information into hormones, and ultimately produce a response to whatever is happening. So they don't have a cardiovascular system, they don't have a nervous system, but we are all controlled by hormones, both plants and animals, and both of them are involved in this calcium to ROS, etc., secondary messengers, which we'll get to a little bit later. Okay. Just don't forget that you will see later we will have primary and secondary messengers, but we'll get to that. Don't stress. Okay. So remember, just basic definitions. Growth is an irreversible increase in cell number and cell size. You cannot go back to being a baby. A tree cannot go back to being a seedling. Even if you cut the tree down, it's still gonna grow straight from those branches into another shoot with multiple meristematic regions. As much as I would like to reverse time and not get wrinkles, it's not going to happen. If you know how, please, I will be the first to pay you money. But yes, growth is an irreversible increase in cell number and cell size. Differentiation is when your meristematics produce cells and then tell them what cells or what tissue types they're going to be. Okay, they have been given their job, they have been differentiated. Also, just bear in mind that some people do grow smaller as they get older, but that's not due to you losing cell numbers and etc. You're losing mass because you're growing old, you're shrinking back into yourself, but you're not going to become a baby. Don't get confused. Okay. So, we get this idea of polarity. Now, remember, when you grow a seed, it's almost as if the seed can tell which side's the light, which side's the gravity, etc. And polarity is important. Remember, tropism is what stimulates and the stimulates such as gravity and light will initiate polarity. Say that again. The stimulus such as gravity or light is what will initiate polarity. So once polarity has been established, it is very difficult to reverse that. So for example, when you're at this stage, it's still possible for you to turn the seed around and have this flatten out again and then start growing the roots down here. But when you start getting to this phase, this is a fungus, by the way. This is a fungus zygote. When you start getting to this stage, you've got already, because remember, the apical meristematic regions are formed during embryogenesis. So once we've got this stage here already, we've got an established shoot apical meristematic region up here and a root apical meristematic region down here. It's already established. Even if we turn this upside down, so I'm going to flip one slide ahead. This is a willow, and they took cuttings from the willow, and they planted it in water, and they planted this one right side up, and this one upside down. See here, the meristematic regions are established. They're established during embryogenesis, and because they're established during embryogenesis, even if you turn it upside down, the roots will still grow there and the shoots will still grow there. The only difference is now, so they hung it up by strings. The only difference is now the shoots are dictated to by the sun. So the sun's up here, so they're growing towards the sun, and the roots are growing down towards the ground. So polarity, once fixed, is not easily reversed. Okay. All right. Now, on YouTube, I have put the this is a movie. The acting is brilliant. The triffids are amazing, and the movie is actually on YouTube. Let me know if you want me to put the YouTube movie on Moodle if you want to watch it. But I have put the trailer on Moodle for you to go and watch and enjoy. But pretty much the Day of the Triffids, it is based on a book by John Wyndham. It's a 1951 post-apocalyptic novel, and in the book we follow Bill Mason. He's a biologist, and he's working on the triffids, and they're a type of plants that have been manipulated and genetically played with, etc. And during his laboratory work, while he's down in the dungeon, there is a beautiful meteor shower. And in the book, people who watched the meteor shower went blind. And then all of a sudden, the triffids escaped from the lab, and then they were carnivores and they went hunting of people, and Bill Mason is like, what the hell? And he goes after trying to capture them again. But in the movie they've changed it around a bit, and the plants actually come from the meteor shower, so they're aliens, and they're literally plants that walk around eating people. It's amazing. I mean, I can't describe to you guys. This is like on Sharknado level of movies. It's really well done for that time. It was made in 1963, the original. There are a few others out at the moment. I think the latest one was 2000. But ultimately, Candice, why the hell you want me to watch a movie about walking plants that eat people? Because plants do move. All plants move. but they don't pull themselves out of the ground and chase you and eat you, like the triffids do in the movie. But we have to understand, as plant physiologists, we have to understand how plants move, what dictates a plant to move, and ultimately, how. How does the plant move? What makes the plant want to grow towards the sun? And if it is growing towards the sun, how? Well, how, you ask? It's called sensory and communication. And it's remember, plant growth hormones. Plant growth hormones, plant growth regulators. So you'll see down here at the bottom, if you see PGRs, it stands for plant growth regulators. And remember, plants sense light, gravity, pressure, attacks from enemies. We've spoken about the giraffe. We know that this is a thing that happens, and the responses to responding to those are in the genes. The genes have responses to this. Remember, the genes dictate how everything's going to happen. Remember, when you're looking here at this stage, you've got this idea that polarity, that the light and the gravity establish it. But when you start getting at D, hormones are released. Hormones are then sent to the genetic codes and said, all right, we officially can tell you that this needs to establish the root apical meristem, and this needs to establish the shoot apical meristem. The genes are switched on, and once switched on, cannot be switched off in certain cases. For example, apical meristems. Other genetic coding can be switched on and off, but that will be determined by the hormones that are released and whether that gene is able to be reversible. Some cases, not all. In the case of apical meristematic regions, not so much. So you have this idea that something is going to stimulate, and then the plant's going to respond, and it's going to respond because hormones are going to be released. So something's going to stimulate, hormones are going to be released based on what the stimulation was. Those hormones are going to go then to the genes, and they're going to activate the genes or switch the genes off, switch the genes on or off. So a plant growth hormone or a plant hormone is an organic compound produced in small amounts in one part of the plant, transported to the targeted cell in another region where a physiological response is caused when a gene is switched on or off. Okay, so there are 3 phases to plant sensory systems. So we're going to take what we've learned from this side, that we have a stimulus, it initiates hormones, the hormones get sent to that area that's been affected, and it switches the genes on and off dependent on the response. We're now going to put in 3 stages of signal transduction. I'm going to say that again. There are 3 stages of signal transduction: reception, transduction, and response. Reception is going to pick up that stimulus. It's going to be transduction or transduced using a hormone, and we're going to get a response that has been activated by switching on or off of a gene to create a certain response. Okay, 3 stages. A receptor that changes in response to a stimulus. It sends a signal transduction pathway to transform the stimulus from accepting it to a hormone that hormone is then sent to the specific targeted genes, and it activates whatever it needs to activate. It's biochemical or cellular responses. Take a moment, replay this, because this is critical. You've got to understand the connection of a stimulus being transduced into a hormone, and that hormone traveling to the targeted area, switching the targeted gene on or off to get the cellular response needed to deal with the stimulus that has been that is affecting the plant. Just bear in mind, I have mentioned before that that Arabidopsis is our model plant in terms of molecular aspects. So most of what we discuss is based on Arabidopsis, but because this is the model, we just translate it to other species. So remember your 3 phases: reception, transduction, and responses. So you have the cytoplasm, you have the plasma membrane, the signal molecule. So you have the signal molecule comes and affects this receptor. The receptor wakes up and is like, oh, something's affecting me. And then it sends through the signal transduction pathway, it sends a hormone to the appropriate gene, and the gene activates a cellular response. So for example, this is a tomato plant. And the tomato plant over here is what was done to this tomato plant was it was grown till a nice healthy seedling like this. And then what we did was every day this one got 0 brushing. So we bumped it, lightly touched, 0, 10, or 20 times a day for each day consecutive. So each day this one got 20 brushings gently, each day this one got 10 brushings, and each day this one got 0. So you just gently, you petted it like you would pet a cat. You just gently brushed it. You'll see that this plant is very tall, very thin, nothing's really affecting it. This one, 10 per day. It's a little bit more thicker, a little bit more shorter, broader. This one's very short, very thick around here. So we've got this response to wind or touch. So what's happened is we've had a stimulus. Nothing's happened here. This one, the stimulus was read, bending and brushing of this one. It was received by a receptive receptor. It went through transduction where it was processed and a hormone was released. And then the response was, hey, don't concentrate on growing upwards, concentrate on secondary growth, increasing your girth, because increasing your girth is going to make you stronger to withstand this touch or wind. I hope that makes sense. So remember now, we've got 3 stages: reception, transduction, and response. And it's in the transduction phase where a hormone is released to go to the specific gene to activate on or off, activate or deactivate. So we have to quickly understand our hormones. We have to understand that now, please. It says 5 main classes. This is 5 main classes that I would like you to understand for this module. By no means is this the only 5 classes of hormones. The plant has multiple hormones that happens. I just expect you to know these ones. Ascorbic acid, Please, everybody, take a note. Homie Simpson, take a note. Ascorbic acid is the same as abscisic acid. It's the same as ascorbate. It's all the same. Ascorbic acid, abscisic acid, as well as ascorbate. Same thing. So this is the same as I'm even getting confused. Okay, let's carry on. Take note. Same thing. So the 5 classes I expect you to know is ascorbic acid, gibberellins, cytokinins, ethylene, and auxin. I cannot stress to you guys enough. Put a star next to auxin. Put exclamation marks next to auxin. Go read up more about auxin. Auxin is incredibly important, as you are later going to find out. We have our definition again. It is a compound that is synthesized in one place where the stimulus is received, and it is moved to another where it has to have the effect. It's a little bit more simple structures when compared to animal hormones. And just remember that one plant hormone has many jobs. So one stimulus, stay with me here, one stimulus can initiate multiple hormone responses, and one hormone can cause multiple responses as well. Plant hormones do many jobs. It's not just one job at a time. So for example, our auxins are really involved in cell elongation, root and apical meristematic regions, which is going to become very important later on. Gibberellins are so important in germination. It's not here, but very important in germination process where the seed actually activates and we start getting the growth of the seed from the radical from the seed. Cytokinins are so important in cell division with that meristematic region. And, sorry. Auxin and cytokinin work in a ratio together. So depending on how much auxin and how much cytokinin will depend on quite a few number of things, which you are later going to see in tissue culture. Remember, tissue culture, the growing of plants in aseptic conditions. And as biologists, we really manipulate the ratio of cytokinins to auxin to get us what we want. Ethylene is a gas. Remember, your mom will, some moms will put ripe bananas with other fruit. If the other fruit is not ripening fast enough, or they'll put it in newspaper to trap the ethylene gas to cause ripening faster. Ethylene is also a very important hormone involved in senescence, which we will get to, because remember, senescence is a programmed cell death, drawing in the nutrients from that plant cell, from that leaf cell or leaf, and then when it gets old, ethylene is released, that hormone, so that it falls off of the plant. Also in fruit ripening, it is very important. And ascorbic acid, which is so important in maintaining health, protecting against rust, bud dormants, fruit maturation, and it also shuts the stomata. Ascorbic acid will be what is released to close that stomata very quickly in a drought or whatever conditions that is going on. So once again, our 3 stages: reception, transduction, and response. We have a stimulus, we have signaling transduction happening here, and we have a response. Just remember, the cells that sense the stimulus is not the cells that respond to the stimulus. The cells that sense the stimulus is not necessarily responding to the stimulus. What's responding to the stimulus is the genes that have been turned on or off because of the hormone released, and the hormone that was released because the stimulus told it to release it. So just a few definitions that I don't want you to get confused with. Transduce, so transduction. So from the stimulus, we get a response, and that is to convert energy from one form to another. So whether it's touch, chemical, mechanical, whatever the case may be, that stimulus is changed to a chemical or hormone response. There is a conversion of energy from one form to another. That is the in between the reception and the response. We have this transduce phase, transduction phase, where we have energy converted from one form to another. Remember, transcription is the process of mRNA formation from a DNA template, and translation is the process of the protein and peptide synthesis from the mRNA. So what's happening? Stay with me. We have a stimulus. We have a reduction. We have energy being converted from one form to another to a hormone, which initiates a response in the genes. We have transcription happening in the genes, and then translation is going to form a protein or a peptide synthesis, which is going to help cause our response. Okay. So here is a nice picture of signal transduction. Remember that this process can lead to one stimulus, can lead to multiple responses, and one hormone can also cause a lot or can have multiple jobs. So just bear that in mind. Okay, so signal specific receptors. So this receptor, so on your plasma membrane, you'll have specific receptors that will respond to specific signals. Signal specific receptors. You have one for light, you have one for touch, you have one for ROS, mechanical damage, etc. Signal specific receptors, because it'll be that receptor. So you have a signal, you'll have that receptor causing a protein catalyst, ATP being used here, not terribly important. Just bear in mind that you have phosphorylation occurring here. And then we have the protein being channeled through certain ways, whether it goes to the DNA, whether it goes to the mRNA, or whether it goes to a change in the ion flow through a pump. Remember that ATP, remember on our photosynthesis, our life reactions, we have that ATP synthase. Remember, we also have ATP synthase where we have positive H ions being pumped in or out. And this is going to be very important when we get to the venous flytrap. But yes, you come here, you get signaled by a single specific receptor. We have a bit of energy being used here, and we can either go straight to the DNA to activate or repress. We could go straight to the mRNA to activate or repress, or we can go to the change of ion flow through that ATP synthase enzyme. And this is really going to affect mechanical movement very much so. So these routes generally are going to be physiological or morphological and biochemical. This one is really going to be a mechanical movement. So I'm sure you guys know of a venous flytrap where it shuts closed. This is how it does it. It does it with the aid of this ATP synthase. So don't get confused. We have our three stages: reception, transduction, and response. This is just showing you that the response can occur one of three ways. It can go to the DNA and activate or repress. It can go to the mRNA, activate or repress. So the DNA, because okay, let me step back for a moment. Let me go through this. Remember, your DNA is going to transcript and your mRNA is going to translate. So when it comes here, if it goes to the DNA, it's going to transcript. It's going to activate or repress transcription. If it activates transcription, it's then going to go to the mRNA, which is going to translate it to give the response. If it goes straight to the mRNA, it's going to activate or repress. Now, it can only go straight here if the DNA has already transcription. So if there has already been transcription, the response or the hormone will go straight to the mRNA. If there has been no transcription before for this gene, it will have to go to the DNA. If it is a mechanical response, if it is a physical response that such as the leaf shutting close on the Venus flytrap or the forget-me-nots, it will go straight to this ion pump. So you have a signal coming in, you have a specific signal, specific receptor cell. We have a bit of energy being used so that the sensory cell can produce a hormone. That hormone is going to specifically go to either DNA to activate or repress transcription, to make the map or to make the recipe. Or if the recipe is pre-existing, it will go to the mRNA to activate or repress. And if it is a mechanical process where the leaves affects the quick and sudden opening and closing of leaves, such as the Venus flytrap, it will go to the ATP synthase pump, which pumps ions. Okay, so recipe not in existence, it'll come here. Recipe already in existence, it'll come here. Quick mechanical closure or opening of cells, it'll come here. Okay. So here we go, electrical signaling. So when you come to this stage here, when it decides to take this route, it is called electrical signaling. And it is touch sensitive. The mimosas, sundews, Venus flytraps, etc. Now, all this is saying, it's saying that on this level, we have a negatively charged plasma. We have a negatively charged exterior. So the plasma membrane up at the top here is negatively charged. And we have ATP, we have H positive ions in here. So this is negative. This is generally positive almost. And then we have a stimulus coming and touching. The receptors read that, those signal specific. It changes it. Remember, it transduces from one energy to another energy. So it changes it from mechanical to electrical stimulus. And this is because the hydrogen ions will be pumped through quickly, and it will change it, and it will quickly change that voltage. It actually has a voltage energy to express as the state of the cell's interior relative to the exterior. So remember, the cell interior is negatively charged. we have those hydrogen positive ions being pumped in quickly, and then we have that change from a mechanical to an electrical. So here we have the Venus flytrap. The receptor cells are at the base of these hairs. You can see here, there's little hairs. And what happens if the fly comes and he lands and he starts eating, and then he bumps this one and he starts eating, and then he bumps one over there. If he bumps two of those hair within 20 seconds, so what happens is he bumps the first hair. The receptor cell picks it up and starts preparing. It starts getting those positive ions close to the ATP synthase. When it touches it again, it quickly pushes all those positive ions into the negative one. That negative energy is converted from mechanical to electrical, and all of a sudden, did you hear that? It shuts closed, and the poor fly is in his prison. So we have this idea. So it goes from negative to positive, back to negative. You've got this. So remember, you are negative here, positive there. The fly, he lands, he bumps. All of the positive ions start gathering here. If he bumps the next receptor cell within 20 seconds, those positive ions are going to shoot through, and you're going to quickly go from a positive to a negative, and that electrical energy, that mechanical stimulus is transduced to an electrical stimulus, and it shuts close. So this is just a graph to show you so this is within 20 seconds. The fly comes, you get a touch, and the plant starts counting down. 1, 2. It starts collecting all those positive ions. This is what it looked like. It connects, it connects, it connects. Then it starts preparing, and then all of a sudden you've got another hit, and you've got flow and shut. If it was wind, for example, or it was a leaf that touched it, remember, it's got 20 seconds. So after 20 seconds, it kind of resets itself to normal. So remember, the stimulus is on the end of those hairs. It gets touched. The receptor cells, it gets transduced or transduction from mechanical energy to electrical energy. And then the electrical energy sends an action potential, and in response, rapidly changes the turgor pressure that moves the leaf and closes the trap. Now, there is a YouTube video by David Attenborough. It really explains it. So remember, so all you need to remember is how the stomata cells operate in the leaves, about turgor pressure. You should know turgor pressure. You should know everything about it. So that action potential rapidly changes the turgor pressure, and you can think that in order to shut close, the turgor pressure has to drop, and the cells have to collapse. So it will shut close. Now, this YouTube video is David Attenborough showing you how the fly gets trapped. Very good. But this YouTube, This YouTube video will explain the turgor pressure of the Touch Me Not plant. So the Touch Me Not plant works very similar to the Venus flytrap. And the Touch Me Not plant really does that YouTube video down here, all on Moodle, don't worry. This YouTube video shows you very clearly how the turgor pressure changes it. The only difference is the Venus flytrap, the turgor pressure is the response, the rapid response of it. So remember, those hairs get touched. Within 20 seconds, we've got positive ions being, so we're going from a mechanical, from physically touching it, from mechanical to electrical. We're getting this shot of electrical current coming through. It's affecting the turgor pressure, or the turgor in the water pressure in the cells, and we're going from turgor to flaccid, and shuts closed. Okay, so if you're a bit confused, watch this mimosa plant YouTube video. It'll explain the turgor pressure very nicely, but instead of mechanical being transduced into chemical by the mimosa plant, we have mechanical to electrical for the Venus flytrap. So the Venus flytrap, mechanical to electrical, changes the turgor pressure. Mimosa, mechanical to chemical. So the mimosa works it's called the touch me not plant. So if you actually go and touch it, it quickly closes its leaves, and that is from mechanical. You actually touching it, it releases a hormone, that transduced from a mechanical to a chemical. That hormone is released. The hormone quickly reduces or takes out the water, and the plant collapses, or the cells collapse on itself, causing the mimosa to close up to protect itself. Touch sensitive plants. So I'm going to end there, because we're now going to go on to tropism. So we understand very clearly that signal transduction has three phases: reception, transduction, response. We understanding that transduction phase is really from one energy to another energy. Hormones are being released in response to it, and we had a look at two examples of the Venus flytrap using electrical and the mimosa using chemical. So just that's all you have to know. You have to understand those three steps. Do understand this graph, this diagram. Understand that it's signal specific receptors. We get transduction, and we can go into three areas. Remember remember our first example with the tomatoes being touched, that will take a route of either DNA or mRNA, whereas this is going to be the mechanical physical shutting ones. So remember, if the recipe is already in use, it will go to mRNA to switch on or off. If the recipe has never been created, it'll have to go to DNA first. Okay, so I'm going to end off there for this section. We will start off with tropism in the next one. So now we're gonna assume as we go forward, you understand those, and instead of every time saying a receptor cell and then transduction and then a response, we're gonna just quickly go straight into tropism. Okay, I'll see you guys in the next video.